Morning. Welcome to this session for the finalists of the Special Gordon Bell Prize in COVID-19 based HPC research. The committee identified three outstanding nominations, and they will be presenting their talks this morning. The first one is called GenSLMs, Genome-scale language models reveal SARS-CoV-2 evolutionary dynamics. And to present this talk is
There's something of an irony in these technical difficulties going on at the supercomputing conference.
A little bit, yeah.
Okay, looks like we're all set. Yeah, that's okay. All right. Good morning, everyone. Looks like screaming is off the table, so we're all clear there. Good morning, everyone. My name is Kyle Hippie, and I'm a research scientist at Argonne National Lab. And today I'm presenting our work entitled GenSLMs, which are genome-scale language models that reveal SARS-CoV-2 evolutionary dynamics. So as most people know, the goal of SARS-CoV-2 is to bind to a host cell and then use RNA replication machinery to produce as many copies of itself as possible. As the virus replicates, it has the possibility of picking up mutations, which can alter the ability for the virus to enter the cell or affect the severity of the infection. The standard way of analyzing these mutations is through phylogenetic analysis, which is shown here. The colors represent different strains of the virus and provide information about its genetic lineage. Mutations that cause an increased infectivity or present with more severe symptoms are referred to as variants of concern. SARS-CoV-2 has a relatively slow mutation rate of roughly 2 mutations per month due to complex proofreading machinery. And of these mutations, only some can give rise to a variant that has more transmissibility or increased severity. Identifying variants of concern has driven much of the public health response, but it is a tedious and resource-intensive process that can take months for a highly skilled team of experts to conduct. Thus, there is an urgent need for scalable, automated, and reliable tools for identifying variants of concern. Achieving this could lead to establishing a continuous watch list of variants for concern, more efficient management of pandemics, and faster solutions from vaccine and drug development efforts. Variants of concern arise through complex interactions between the virus and the host. One of the most popular candidates for study is a spike protein, and while this is a crucial part of the virus, it's just one of the many interactions between the virus and the host cell. For instance, most therapeutics target the non-structural proteins, while vaccines mostly target the spike. A model that takes into account the entire genome of the virus would allow valuable insights into other potentially vulnerable regions within the virus, and with this information, we could create a continuous watch list of variants for monitoring, model how evolution impacts new strains, and assist development of vaccines and other therapeutics. The success of language models on natural language has prompted people to expand them to protein sequences, which have shown remarkable success in applications such as protein engineering. Protein language information, though, sits 2 levels below the genome level information, as genomes must have DNA, which is transcribed into RNA and then translated into proteins. This means that all of the information encoded at the protein level is also encoded in the genome level, along with valuable additional information, such as post-translational modifications. Furthermore, information contained at the gene level is often carried over within viral genomes and is connected all the way up to humans. Hence, building a genome-scale model could be valuable in interpreting the nature of how SARS-CoV-2 evolves. In this work, we present several genome-scale language models that tackle longer sequences, have some of the largest set of trainable parameters, and are trained on one of the largest genomic data sets, exceeding current state-of-the-art in these categories. However, one needs to overcome several challenges when working with genomes, namely addressing the much longer sequence length and the application of NLP techniques to biological data. Here we present models that can learn biologically meaningful latent representations based on codon-level tokenization, and as a solution to the large sequence lengths, we develop a hierarchical diffusion model that can capture context across long sequences. Lastly, we present an end-to-end workflow that integrates AI and biophysical simulations to identify variants of concern at the genome level. Through these experiments, we created foundation models trained on more than 110 million raw nucleotide sequences, with model scales ranging from 25 million to 25 billion trainable parameters. We present the scaling of these models on Selene at NVIDIA, as well as Polaris at ALCF, along with SARS-CoV-2 specific models trained on the Cerebras CS2 wafer-scale engine cluster. This culminated with 1.63 zettaflops utilized for training the foundation models and achieving 121 petaflops sustained performance with peaks of 850 petaflops and mixed precision performance on Selene. So working towards the end goal of having models that can help us identify variants of concern, we started by trying to train the models directly on the genome sequences of SARS-CoV-2. However, much of the genome is conserved between these genomes, with less than 1% of the genomes containing meaningful mutations. The solution then to our problem was to train on a diverse set of 110 million genomes to bootstrap the representative power of the models, and then fine-tune them on the SARS-CoV-2 genomes, periodically as new data enters our pipeline. For our detection workflow, we use the genome models to calculate two scores, semantic similarity and the sequence log likelihood, which are components of the immune escape scores and fitness evaluation scores. From there, we turn to the biophysical side and use OpenFold, which is an open source variation of AlphaFold, to fold the spike proteins and extract epitope alteration scores, as well as spike binding affinities to the ACE2 receptor. These scores then combine to give us a final variant of concern score. Additionally, we would like to leverage the ability of the generative capacity of these models to detect potential variants of concern. To do this, we use a hierarchical diffusion model to generate synthetic genome sequences and feed them through the detection workflow to get their variant of concern score. Starting at the top of the workflow, let's take a look at the genome-scale language models. Our genome-scale language models follow a typical language model architecture where the sequence gets fed into an embedding layer that's followed by numerous transformer and attention layers. One of the main challenges presented by these attention layers is that their complexity grows quadratically with the length of the sequence. This has traditionally been a challenge for large language models to capture context beyond a certain sequence length. From the transformer layers, we can extract semantic embeddings or decode our latent representations back to the codon language. We provide various sizes and configurations of these models, from 25 million trainable parameters up to 25 billion trainable parameters, and have open sourced them for the scientific community. These models were trained on a diverse set of 110 million gene sequences in order to overcome the challenges mentioned earlier about the large areas of concern sequences in the SARS-CoV-2 genomes. And this resulted in some of the largest biological models trained on genome nucleotide sequences to date. Being one of these first genome models, we must come up with a new solution for evaluating their performance. So we started with a common NLP metric, and we look at the perplexity of these models on a held-out validation set of data. Perplexity can be thought of about the number of guesses the model needs to predict the next token given some input, and a perfect score would be 1, as this means that the model can guess the correct token on the first try. When evaluating the perplexity of these models, we generally see a decrease as the total number of trainable parameters increases. The 2.5 billion parameter model achieved a perplexity of around 1.25, and most of the results following this are based off of this model. So due to the time and resource constraints involved in training the 25 billion parameter model, we were unable to train this model to convergence. But using the CS2 wafer-scale cluster, we were able to achieve convergence when training on the full SARS-CoV-2 genomes, and we'll present those results shortly. So before we talk about performance, it's worth noting a couple of things about our infrastructure. Our main targets for the system were Polaris at ALCF, Selene at NVIDIA, and Perlmutter at NERSC. Polaris and Perlmutter nodes are composed of 4 40 gigabyte A100 GPUs, whereas Selene nodes have 8 80 gigabyte A100 GPUs. Our software engineering structure is built on Hugging Face library with a PyTorch Lightning backend utilizing Deep Speed 03 optimizations. This allowed us for rapid development and testing of different model architectures, as well as quick deployment to new systems. We were able to deploy the code to Selene in a matter of hours using standard development environment provided by NVIDIA in order to get performance scaling measurements. Performance is measured using the Deep Speed profiler and was correlated with NVIDIA NSYS. Here we look at the performance of these models across the two platforms, Polaris at Argonne and Selene at NVIDIA. So we see as model size increases, the throughput decreases, with a 25 million model achieving roughly 10 to the 4 samples per second, and the 25 billion parameter model achieving roughly 10 samples per second. Conversely, as model size increases, we see better scaling due to the increase in model computations that allow us to overlap these computations with communication and achieve higher overall utilization. To determine the reason for the decrease in scaling performance beyond 512 GPUs, we compared deep speed 0.3 optimizations with standard DDP in an experiment not shown here, and saw near perfect scaling as compared to deep speed 0.3 due to the use of all reduce in 0.3 versus all gather in DDP. Additionally, due to the 80 gigabyte cards available on Selene, we were able to achieve 2 times throughput when compared with Polaris, which is attributed to the doubling of the batch size. For the performance runs on Selene, we achieved mixed precision performance of 121 petaflops sustained, which accounts for the end-to-end training scheme of data loading and model initialization, and a peak performance of 850 petaflops, which is measured as a single layer across all GPUs. In total, for the science runs needed to pre-train these genome-scale language models, we used 1.63 zettaflops in mixed precision. Now that we have trained models, we need to evaluate if they can capture biologically relevant information. To do this, we embedded a subset of the validation dataset and painted each point in the genome with relevant biological information to look for clustering and gradients that suggest semantic meaning within the latent space. Shown here, we see clear clustering and gradation based on the length of the genome sequences, as well as the GC content, which is an indicator of the relative stability of the RNA sequence. This indicates that the model has found a rich representation for these sequences. To further evaluate the biological meaning encoded in this model, we examined a specific enzyme called malate dehydrogenase, which has 2 unique isoforms. The model has placed these isoforms in unique clusters that highlights the structural differences, of which it has no pre-existing information, and it preserves the meaning of these other biological relevant metrics across, in addition to respecting the structure. So after the foundation models are trained, we look to fine-tuning these models on the SARS-CoV-2 open reading frames. For this presentation, we're looking only at models that have seen data from the first year of the pandemic, so keep in mind that this model has only seen the alpha and beta variants coming from the first year of the pandemic. So when we embed these genomes from the entire data set that include data relevant to the Delta, Omicron, and other variants seen in the second year, we see that not only does biologically relevant metrics stay clearly clustered and gradiated, but the model can also cluster on genomes based on the variant type, including variants it has never seen before. Additionally, since there is no notion of variance in the training data, any semantic clustering based off the evolutionary traits is purely a result of the learning capacity of the genome-scale language models. So looking a bit more closely at the variants, this image represents the information encoded by the model when looking at the iota variant. Around the outside, we see the open reading frames, and directly beneath, we see the proteins that they encode. In the blue color beneath the protein label, we see the log likelihood of the particular sequence, where deeper blue represents a more likely sequence and lighter blue represents a less likely variant. Finally, the connections in the middle represent evolutionary connections across the different reading frames. Looking next at the beta variant, we see that this variant is less likely to occur given the lighter blue inner ring, and when we compare the evolutionary connections between the spike protein and the iota variant and the beta variant, we see not only connections inside or across open reading frames, but we see different connections among different variants. Since these connections are different across the variants, this indicates that the evolutionary cross-coupling provides insights into potential phenotypic differences. Although we have not related it back to phenotypic expression, this model lets us probe interactions, which may be useful for studying emergent mutations across the spike and other proteins. Due to the time and compute required to fine-tune these larger models on the SARS-CoV-2 data, we turn to the CS2 wafer-scale engine cluster to accelerate training and time to solution for the SARS-CoV-2 genome-scale models. The CS2 wafer-scale engine cluster is one of the largest integrated chips in existence and is designed to perform common deep learning operations quickly and efficiently. The CS2 allows users to run jobs as you would on a regular cluster. You submit jobs and the appliance manages pre-processing, streaming, I/O, and device orchestration transparently using the SwarmX network interface. The platform provides linear scaling up to 16 CS2 machines and is designed to enable training of massive models using only data parallelism. Using a four-node Cerebras cluster, we train from scratch until convergence two models, a 123 million parameter model and 1.3 billion parameter model. We achieve greater than 96% validation accuracy and took less than half a day to converge on 4 CS2s. Evaluating the model's latent space, we see similar trends that it's able to encode biologically relevant information and is still able to cluster genomes by variants. On the performance runs, we were able to show near linear scaling to a cluster size of 16 CS2s across 3 model sizes, ranging from 250 million parameters to 25 billion parameters. Next, we shift attention to look at leveraging the generative powers of the GenSLM models. In order to address full genome sequence generation, we needed a method that could handle the long-range sequence dependencies present in the SARS-CoV-2 genome sequences. Our solution is a hierarchical diffusion model that can capture long-term dependencies in sentence-level representations that allow the language model to learn long-range contacts while overcoming the problem of extremely long sequences. The hierarchical diffusion model uses a standard Gaussian noise and denoising training scheme and is trained on word-level embeddings coming from the 2.5 billion parameter model. This enables more accurate genome generation than achievable with standard language model generation algorithms or sequence generation algorithms. The hierarchical diffusion model can generate synthetic sequences that make biological sense. These sequences coming from the model more closely mirror the distribution of open reading frames found in the real-world data when compared to standard generation. And when looking at the overall distribution of sequences, we can perform phylogenetic analysis and see that sequences coming from the model integrate into existing lineages among the major variants observed from the pandemic. Lastly, we turn to the final portion of the workflow, which incorporates biophysical experiments into the variant of concern scoring. As stated previously, synthetic genomes fall into these existing lineages. Dots in orange represent generated sequences, and bars in gray represent real-world lineages. We consistently see that generated sequences fall within existing lineages as well as extending them. However, if we try and compute on all these generated and sequenced genomes, it becomes computational intractable to try and fold them and simulate them, as the folding jobs take on the order of 30 minutes per sequence, and MD simulation can take hours to reach stable convergence. To reduce the number of sequences we perform these experiments on, we filter based on the number of phylogenetic neighbors from each lineage and cluster these to get representative samples from each lineage to perform these expensive computations on. So this is a small example of generated sequences that have been folded and put through our MD simulation pipeline to get their binding score with the ACE2 receptor. In the figure here, we see that the difference in binding score for generated sequences represented in each individual plot when compared to the major wild-type variants labeled across the bottom of each plot. There exist variants that have a more tight binding score with the ACE2 receptor than many wild-type variants. This further reinforces the power of the hierarchical diffusion model to capture not only the evolutionary information, but maintain biophysical properties that are not present in the training data. This is ongoing work as the computation required for the MD simulation. Convergence is large and will require further algorithmic advancement to run at the scale of full genomes being sequenced by testing facilities. But when we look at a specific generated example, we've overlaid the results of the MD simulation with the synthetic sequence when compared to the wild type result of the simulations. And in yellow, we can see the mutations of the sequence and how they change the binding region of the protein in order to enable a more stable interaction between the SARS-CoV-2 virus and the ACE2 receptor. So, in summary, our work presents one of the first large language models trained on genomic sequence data and represents one of the largest biology models trained on one of the largest corpora of gene sequences that exists. The models we show can capture a hierarchy of biological information from metagenomic data down to gene product structure. Through the process of creating these models, we achieve a total of 1.63 zetaflops with sustained performance of 121 petaflops in mixed precision and a peak of 800 petaflops of mixed precision. We showcase the CS2 wafer-scale engine cluster that can achieve model convergence in less than a day and scale linearly up to 16 nodes. Lastly, we contribute a novel end-to-end integrated workflow that enables analysis and visualization of variant of concern monitoring. This work has implications beyond biology, to biology beyond just SARS-CoV-2. Our genome-scale models offer novel insights into organization of genes. They capture more information than a standard protein language model encoding, and they can be applied to problems beyond COVID-19, such as the annotation of unknown genes and proteins, gene-protein engineering workflows, and vaccine or therapeutic design. Lastly, our work contributes a distributed workflow that enables the characterization of variants of concern at the genome level. It showcases the integration of and chaining of AI approaches in natural language and structural biology domains, as well as the future work revolving around the integration of neural operator models to accelerate the biophysical molecular dynamic simulations. Finally, we present the ability to perform genome-scale inference in conjunction with biophysical methods at scale. So, of course, this work cannot be done without the contributors of many people from organizations including NVIDIA, Cerebras, our collaborators at Argonne National Lab, University of Chicago, Northwestern, and University of Illinois Chicago. And we'd like to extend a special thank you to the visualization team and data storage team for enabling us to share this work with you today. Lastly, we'd like to acknowledge our funding sources as well as the computing time that we were able to reserve. And we would like to thank all of our colleagues for their assistance at all steps along this journey. At this time, we would like to field any questions or comments, so thank you for your time.
Hello. Yeah, so thanks for a really cool talk. So as we've seen, pandemics are really bad for like all of society. So, and we're thinking about how, you know, we can prevent future pandemics. So with that in mind, how simple do you think it might be to like generalize this workflow so that at some point you could basically just take any kind of virus genome, just plug it in and then get all this data?
Yeah, absolutely. So that's the goal of this platform when we started. When we found the opportunity to create these foundation models, we saw that this could extend beyond just the SARS-CoV-2 pandemic. So we can take our foundation models, which we've shown can capture relevant biological information, and we can fine-tune them on whatever virus or whatever pandemic would arise in the future, and then we can directly adapt this workflow using these fine-tuned models in the same way that we showed here. So it's a very quick time to solution in order to adapt this workflow to other pandemics that might arise.
Hi. So the use of the Cerebras systems is particularly interesting, and there's some hints in the paper as to comparatives between how the CS2 systems performed relative to the really massive supercomputers that were also applied. Would you care to elaborate a little bit on that and prospects for the use of experimental chips like the Cerebras systems?
Yeah, absolutely. So the CS2 system allows for really fast time to solution. So as we showed, we were able to train from scratch on the SARS-CoV-2 genomes and reach convergence in less than a day. So this provides a really unique opportunity to continue or to keep pushing the next step of this workflow without maybe necessarily having to train large models to convergence on other platforms. So this enables very quick time to solution for this type of workflow and workflows that we would see in the future.
Yeah, my name is Dennis. I'm from Argonne Leadership Computer Facility. And so first of all, thank you for the talk. It's very great to see that application of transformers in the science with real success. So I would like to ask about performance. So in Megatron LM paper from NVIDIA, it was shown that basically this approach, I mean, when you have different types of parallelism, outperforms the redundancy optimizer which DeepSpeed is using. So why did you use DeepSpeed? And second thing that they shown that it's very important to use different types of parallelism. Even with that, you can reach maximum efficiency. So which types of parallelism did you use and how you tuned to reach maximum performance?
Absolutely. So as to the first question, one of the major concerns when we were starting this project was the ability to plug in different model architectures and swap them out very efficiently. And that was something that the Hugging Face platform with PyTorch Lightning and the DeepSpeed provided for us. So one of our future works is to see how we can optimize this further using other optimizers such as Megatron or wafer-scale, things like that. And see how we can further push the envelope for the size of these models to get better performance on these new exascale systems that we'll be seeing coming online. And then the second question, sorry, you might have to remind me really quick.
Yeah, about different types of parallelism and how did you tune to reach maximum performance?
Yeah, so I was not directly involved in that process, but we'll reach out to you after this, and we'll connect you with people who can point you in the right direction on that information.
Yeah, if I may love, can I ask small and ask question? In transformers, there is a problem that when it's super huge model, the bigger model is not always the better. So it's very important to use special parameterization techniques. So which techniques did you use? What did you find useful? And
Yes.
it's connected with your probably problem that with 25 billion parameters model, you couldn't train it.
Yeah, absolutely. So first, we haven't been able to perform hyperparameter optimization on the 25 billion parameter model. So we don't think that we found the right connection between learning rate and other metrics to reach convergence. So we don't actually think that that model has converged. But we're definitely looking into working with performance people in order to increase the throughput of that model so that we can experiment with some of those other metrics that you mentioned. But right now, all we have is model sharding and parameter and optimizer offloads onto the CPU and NVMe.
Yeah, hi, thanks for the talk. So I have two questions. One is, do you think you can use your model to predict new variants and the response to vaccines? Because we know that people take 2 vaccines and boosters and still get COVID. So I think having a model that can predict when a new variant will evolve and whether it will respond to the existing vaccines would be really cool. So I'm wondering if you can adapt this for that. And the second question is, you showed some binding energy predictions from simulations. Have you experimentally verified some of those values?
Awesome. So first question. So we are able to show that with our hierarchical diffusion model, we can extend the lineages that are currently existing in the pandemic strains and things like that. So one of the things that I didn't get a chance to mention was one of the scores is called an epitope alteration score. So the epitope is the region of the spike where the antibodies bind to it. So we do actually take into account the ability for vaccines and current antibody responses to bind to the spike. So this is currently in the workflow, and we'll be looking to kind of leverage this further in how we can model this in other pandemics that may arise in the future. The second question was, sorry, remind me again?
Yeah, experimentally verify the binding energy predictions for the AS2 receptor variant.
Yes. So that is problematic at best. We're performing experiments with the malate dehydrogenase in order to verify that our simulations can correctly identify patterns that emerge in malate dehydrogenase. So we haven't been able to confirm experimentally with the SARS-CoV-2 and ACE2 binding, but we are performing experiments on, or like wet lab experiments on the malate dehydrogenase to verify some of the biophysical side of the workflow.
Thanks.
Let's thank Kyle and thanks very much. Thank you. Can we bring up the presentation for the